I had hoped that you could have done 2 of them. I think 2 of you attempted 2 of them, which was good. Let's go through the solutions. So one of them was, I thought was an easy question. Compare a hollow fiber blood oxygenator to the human lung. What are the differences? Define the desired properties of an optimized blood oxygenator. So what we can do is do a table like this. So we can say here's the natural lung. Okay, and here's a blood oxygenator. BO is blood oxygenator, okay? So what can we say about this? I'm going to start off by putting up some things here and you can correct me. So 37 degrees Celsius is the operating temperature of, is it okay? Yeah. Is the operating temperature, right? 37, 38 of the natural lung. Should I keep going or? Okay. So what is the operating temperature of blood oxygenator? It's not 37 degrees Celsius, okay? So it's something else. Maybe 25? Maybe 25 degrees Celsius? Okay. I'll start again. That's good. Close this one? Okay. Okay, good. So let's do this comparison. Oh, I'm going to do this again. 1 second. Okay. So comparison of lung and blood oxygenator. Okay, so maybe we do it this way. So here's what we're going to worry about. So temperature. So here is the lung, and here's a blood oxygenator. So lung, let's just say 37 degrees Celsius. Blood oxygenator, I don't know, 20 degrees Celsius maybe, whatever room temperature is, okay? So one of the things that you're going to need is, therefore, you need heating here. Heating for blood return, okay? So there will have to be some sort of heat exchange. That's one thing that you need. Okay, what else? So in the design, if you say operation time, so this is going to be your lifetime, right? And this is going to be about 6 hours. There's a big difference there because the lung is able to regenerate itself somewhat within limits, okay? But the blood oxygenator, no, it's one-time use. And that's a big design concern, criteria there, okay? What else can we say? Sorry? For the blood oxygenator. Yes, yes. So I was going to put it a different way, but you're right. I was going to say the surface area for mass transfer. So here, the surface area of the lungs is maybe 70 meters squared. And for a blood oxygenator is much, much less, okay? Maybe 0.5 meters squared. And so that is why you need that reservoir, okay? Because you need to have a hold-up volume. And a few other things. On this side, when you think about it, when you think of mass transfer, okay, so the resistance is minimal in the lungs. Mass transfer resistance is minimal, okay? You have basically a diffusion distance that's very, very small. In a membrane, actually, the resistance is higher, okay? So that's another big difference. Some other things that I'll just mention, I won't write down. So the lungs are self-cleaning, okay? And they're highly hemocompatible compared to membranes. The membrane structure in the lungs is sensitive to partial oxygen concentration. The blood oxygenator, there is no responsive membrane, okay? In the lungs, you have a very, very short capillary distance. In a blood oxygenator, you have a much longer capillary distance. Another important thing is that we are trying to minimize the priming volume, right? I mentioned that in the blood oxygenator. In the lungs, you're not trying to do that. You have a huge surface area, 70 square meters actually, okay? Because this is all a part of your body. It's biocompatible. So the priming volume, if you like, of the lungs is quite high. And that's fine, okay? But for blood oxygenator, you're adding an extra volume, so we want to keep it as small as possible. We also have to be careful about sterility in a blood oxygenator. So again, it needs to be compact. And then there are a few other things. In the lungs, there's no anticoagulant. You have to add anticoagulant for a blood oxygenator. Blood oxygenators operate at lower blood concentrations or hematocrit. The lungs are about 30, 35. Blood oxygenator about 15. Okay, you dilute it with anticoagulant. And the final thing is residence time. That's the other important one here for design perspective. So here it's long, relatively speaking, and here it's short. So the residence time that you want for the blood in the blood oxygenator is short because you want to minimize interactions with non-biological surfaces, and you want to oxygenate this quickly in as small a volume as possible, so you're having much higher flow rates. In the lungs, actually the red cells pass very, very slowly through these capillaries, maximizing the possibility of oxygen transfer. Okay, that's an important difference. Okay, so I think those are the main things. There are others that you could put as well, but those were the main things I want to talk about there. Okay? The next question then, so if you've copied what you wanted to from that, I'm going to delete that now and move on to the next one. The next question was, I need to find it, hang on. Oh, here, okay. So I'm going to delete this, okay? Let's move on to the next one. Okay, so I said here the concentration of protein A in buffer B is 25 milligrams per liter. So we have protein A in, that's terrible, hang on. Let's try again. So protein A in buffer B, and the concentration I said was 25 milligram per milliliter, okay, 25 milligram per milliliter. You wish to double the concentration of the protein and transfer it into buffer C. This buffer exchange is to be accomplished conducting a 6 times diafiltration. That means you want 6 diavolumes, okay? So we're going to use, we're going to concentrate 2 times, right, to get from, to double the concentration, and then a 6x diafiltration. Or if you like, 6 diavolumes you want to use, okay? The initial volume of the protein solution is 8 liters. So initially, V naught equals 8 liters. You may assume the membrane surface area is, the membrane surface area A is equal to 0.4 meters squared. And the permeate flux, you're told this, J equals 25 liters per meter squared per hour. Okay? 25 liters per meter squared per hour. Now, you are told it's a constant, so you don't need to worry about changes in flux with concentration. How long will it take to concentrate the protein? That's the first question. So, we're going to do concentration first. How long to concentrate? So I think 2 of you had done this. This is really very simple, right? You know the flux, J is equal to what? It's equal to the volume divided by area multiplied by time, which is equal to Q divided by area, right? Q is a volumetric fluid, or volume divided by area time, okay? And I think we know everything because we are told that the flux is 25. We just have to be careful of the units, 25 liters per meter squared per hour. And what else do we know? That is equal to the volume, and the volume is what? Did I write it down here somewhere? What is the volume? Let me see who knows what volume goes in here. The flux is 25 liters per meter squared per hour. What is the volume? What is the area? What is the time? So we're trying to find time, right? That's what we want to know. How long does it take? Do we have a value for volume and area? Anybody? Anybody? No? Sorry? 4 liters, good, very good, yeah. The volume is 4 liters, that's easy, right? That's the amount of permeate. You start off with 8 liters, you're going to double the concentration, you want to take out 4 liters. That's the permeate, okay? It's not the feed volume, it's the permeate volume, good. And the membrane surface area is what? Good. Divided by 0.4, multiplied by T, therefore T is very easy, that equals 0.4 hours, which is equal to 24 minutes. Okay. What's the next part of this question? The next part is how much buffer C is required. Okay, that's very simple. Who can tell me how much buffer is required for the diafiltration? I heard 48 liters, right? So it's 6 times what is your volume. So hang on, are we doing diafiltration first or are we doing concentration first? You wish to double the concentration of the protein and transmit buffer C. The buffer exchanges to be a complex of the initial volume of 8 liters. You may assume this. Well, it could be 48, it could be 24, right? It depends on when you do which you do first, okay? So I will accept either. So it could be 6 times either 4 or 6 by 8. Okay, I agree. And then the last part is, how long does the entire process take? Well, that really depends on how you're going to do it. So I had assumed that you're going to do diafiltration second, okay, because the process will take longer. So anyway, if you do concentration first, you've got 24 minutes here, and then you have to take flux of 25 must equal 24 is your volume. The area was what? Area was 0.4 by T. That's how you do it this way, okay? And if you assume that you're going to do, actually, you really have to do it this way first because we already worked out the concentration part. But if you want to recalculate it and do 48 here, you can, and then do the concentration. It doesn't matter, okay? But then it will just take longer. So there's actually no point in doing diafiltration first. You just waste buffer, it takes longer. Okay? So that question is clear, right? That was a fairly easy question, I thought. Okay. The last one, did anyone look at the last one? Because the last one was the most difficult. And I said if you had a chance, look at it, but otherwise don't worry. So let's go through it because I think it's a nice problem anyway. So, and this would make most sense to chemical engineering students. It says, consider a flat plate hemodialyzer in which blood flows between two membrane sheets, each of which has a surface area of 5,000 square centimeters. The blood and dialysate fluid, which flow on opposite sides of the membrane, flow countercurrent to each other, and the following conditions apply. So basically, we have something like this. Okay, so we have these two membrane sheets like this, right? So that's one membrane sheet, that's the other sheet. And what we want to do now is just take a thin section here, and we're going to call that dx. So this is dx. Okay, and then we are told various things about this. We are told that The blood flow rate is VB, is the blood flow rate, and that's equal to 1.2 centimeter cubed per second. You're told the inlet urea concentration, CB in, all of you should have the question, right, is 1.5, why does it keep doing this, 1.5 milligrams per cubic centimeter. Okay, what else? The inlet urea concentration in the dialysate is 0. So you have dialysate that's clean dialysate, okay? The mass transfer coefficient for urea in blood is Kb. This is the mass transfer coefficient for urea in blood. That's equal to 1.25 multiplied by 10 to the minus 3 centimeters to the negative 3 second to the negative 1. I'm sorry, 10 to the minus 3. How do I erase that? Centimeter per second. And mass transfer coefficient urea in the membrane, so K membrane is equal to 4.00 multiplied by 10 to the minus 4 centimeter per second. And the mass transfer coefficient urea in the dialysate fluid is 0. So, okay, let's think of this now. So here's what we got. We got a problem like this. We're going to have a membrane here. We're going to have dialysate flowing on one side, blood flowing on the other side. We want to remove urea. There are going to be 3 mass transfer resistances. Think of a flat plate heat exchanger. You have a convective mass transfer resistance on the blood side, one on the dialysate side, and one through the membrane. Okay? On the dialysate side, we are saying it's 0 because the urea concentration in dialysate we're going to assume is 0. Okay? It's very low. So we only need to worry about these 2 mass transfer coefficients, one on the blood side and one through the membrane. So that's the setup of the problem. And again, I'm going through the mathematical version. If you're not an engineering student, it will be more difficult, but you can at least see qualitatively what's happening. So qualitatively, we're trying to work out the rate of transfer of this urea from the blood into the dialysate. So what we typically do in engineering is we consider a mass balance on a small amount of a small volume. What we're doing basically is we're saying we're looking at what happens in this small volume, and then we're going to average for the whole membrane, okay? And that's where this integration comes in. So what we say is that accumulation of urea, okay, in this small volume equals whatever comes in minus whatever goes out, right? So equals flow of urea in minus out in the blood, okay, because there's blood flowing into this little area and out. So whatever comes in minus goes out, plus flow of urea, so plus flow of urea in minus out in dialysate. Okay, so at least I hope qualitatively this is making sense. So here is our membrane. We have various sheets here. We have, say, perhaps blood there, and then dialysate flowing counter current to that, so dialysate coming this way. We're doing a mass balance here, and we're saying, well, the accumulation of urea in this tiny volume depends on in minus out in the blood plus in minus out in the dialysate. And we know that in this area, we're going to have urea transferred from the blood to the dialysate. Okay, so that's how we typically set up one of these mass balances. And then we can go ahead and write this then in differential form. And I'm running out of space here, so let's see. I'm going to write here and then I'm going to erase everything, okay? So, because I don't want to spend too much time solving the differential equation, we say VB, that's in the blood. So the accumulation is 0. For those of you that are not familiar with this sort of analysis, in engineering, we typically take a small volume like this, but then we at steady state say, at steady state, there's no change. So there's no accumulation. So basically, this term goes to 0 at steady state, okay? So 0 multiplied by VB, concentration in the blood at X, minus concentration in the blood at X plus delta X, okay? So what's coming in at x and what's leaving at x plus delta x, okay, plus flow in the dialysate, sorry, plus Vd, that's a dialysate flow rate, right, multiplied by C in the dialysate at x minus C in the dialysate at x plus delta x. Okay, so that's all I've said. There's no accumulation here. Whatever is coming into this in the blood minus what's leaving in the blood, what's coming in the dialysate minus what's in the dialysate. Okay, now I'm going to erase all this. And let's start again. Okay. Okay, so we can then put that into differential form, that equation where we have 0 equals negative Vb dCb dx plus plus what? Plus Vd dC d dx. And actually, if you like, I'll ask Professor Jacob to copy this, and you can have my solution also. Okay, so then you can just do some rearrangement of all this, and you will end up with Vb dCb. Remember, Cb is concentration in the blood, dx, which is a function of x, equals Vd dCd dx. And then we do something that I always think is rather interesting. We say dCb dCd equals Vd divided by Vb. And I always think that's funny, but we say the dx's cancel, okay? So they don't really cancel. This is just something that we learn, I don't know, in differential equations. In reality, we're saying they're well-behaved functions and things like this, and so therefore you can treat the derivatives as fractions, okay? But for those of you who are not taking partial differential equations, don't worry about that. Let's just say that this is a way we can come up with this equation to describe that mass balance that we had earlier, okay? All right, and then actually this one turns out to be fairly easy to solve at this stage, because you just integrate and you say Cb, right, minus Cb naught equals Vd divided by Vb Cd minus Cd naught. Okay, and then you can rearrange this. We know what they are. We know that the concentration of urea in the dialysate at the entrance is 0, so that actually disappears. We have values for what it is for Vb naught and so forth, and so we can rewrite this all, and we end up with something like this: Vb divided by Vd Cb minus Cb naught plus Cd naught equals Cd, and that's my first equation, okay? And I can put some numbers into this that we have so far. We know that we know a few of these values, and so we can come up with something like 1.632 equals Vd Cd naught. Okay, that's my second equation, and I'll give you my notes on this. Okay, so let's continue then. For the last part of this. So what we'll have to do then is next consider a mass balance on the blood side only. So we'd consider a mass balance on blood side only. Okay, and what you say then is accumulation of urea in blood, accumulation okay, is going to be basically, there's a slight difference here. It's in minus out in the blood, so blood flow in minus out, okay, that, and then you're also going to subtract whatever is transferred to the dialysate. Okay, so again, if you're not a chemical engineer, this is just the sort of way we do this. Okay, so you do an overall balance, you do a balance on one phase, right? You can't do balance on two phase because that'll give you the overall balance, and then they're not independent. So you can do this, and the important part is that the other one gave you a relation between CD and CB and VD and VB. This gives you a relation, again, you say accumulation is going to be 0 at steady state. That disappears, but here is where you're going to get your mass transfer coefficient coming in, and that then gives you rates of mass transfer. So what we can say is, I'll just go one step further and say that therefore 0 equals negative VB dCB dx minus 2LK, that's the mass transfer coefficient, overall mass transfer coefficient, CB minus CD, okay? For those of you, again, that are chemical engineers, you should be able to figure out how I got to that. I'll put the solution up anyway as well. And then you can go ahead and integrate that and do various things. I won't spend the time here, except to tell you there are a few tricks to it. This is an overall mass transfer coefficient, so 1 on K equals 1 on each of the individual Ks, K1 plus 1 divided by K2 plus 1 divided by K3. This one we can say 0 because that is going to be in the dialysate, which is said was 0. So you have a membrane and a feed side. Okay, so you have to add those and put those in, and you get a number. I'll let Professor Jacob copy this and give it to you. And so you can come with all that, and you'll come up with a second equation that will allow you then to eventually, you have three equations, then you can calculate, solve them simultaneously, and then you can answer the question. And the question was where was the question? Hang on. The question was here. No, I lost the question. Determine the outlet concentration of urea in the dialysis fluid, CD out, and the volumetric flow rate of dialysis fluid. Let's pause a second because, again, I think it's nice to think of this qualitatively. So in a dialysis problem, right, that is really what you want to calculate because you've got a dialyzer, you're running human blood. You can't change the blood flow rate. That's going to be a certain flow rate that has to match the time taken to treat the patient. You can't really change the initial concentration of urea in the blood because that's what you're trying to remove in this case. And you can't change what your outlet is because that's the amount you have to remove. So you've got to match that with the dialysate flow rate and the amount you're removing in the dialysate, right? And that will depend upon your mass transfer resistances, these mass transfer coefficients, okay? So once you got values of those, then you have to figure out what sort of flow rate you're going to use. And so that's basically what this problem is asking you to do. And I'm just showing you, for those of you that are chemical engineers, this should be very, very similar to a flat plate heat exchange problem, right? It's exactly the same, except two things. You have all these Ks instead of Hs, right? You have Hs for heat transfer coefficients. That's one difference. And the other difference is that you might, instead of a mass transfer coefficient through the membrane, you would have a conductivity. And a conductivity has slightly different units, right, to a convective heat transfer coefficient. So you have a K divided by a length or something, some sort of dimension, right? But otherwise, it's exactly the same, okay? That's why I think it's a nice problem because it seems very complex, but if you've done these sort of problems in undergraduate classes, it should be fairly simple. So, okay, the final solution then is that C, this is going to be green this time, CD naught equals 0.13 milligram per milliliter. That's the concentration of urea in the dialysate at the outlet. And what else? VD, the volume of dialysate comes out to be the volumetric flow rate, rather 12.5 cubic centimeters per second, okay? So, for those of you who are not engineers, maybe you can see that the way we're trying to calculate this is you have two flow rates, right? The blood side is fixed. You're asking yourself the question, what do I have to do on the dialysate side to achieve my separation, okay? Also, the membrane area is fixed this time. You can't change that. You've got a dialyzer, okay? And then for chemical engineers, I'll give you the actual full solution, but that's the way you would solve it. Mass balance on the overall system, mass balance on one of the phases. You could have done a mass balance on the dialysate instead of the blood. It's a more complex though because the dialysate you're trying to calculate things there. So it'll become mathematically more complicated, but you can do it, okay? And then you solve three simultaneous equations and you come up with the numbers, okay? So that question I put there on purpose. I thought it was a bit difficult, but on the other hand, it was one that kind of links up with what you probably learned earlier in classes, so I thought it would be interesting. Okay, any questions on all that? So I just have one thing left to do then, and that is to talk about this very last lecture, okay? All right, so share and, oh, someone needs to make me a host. So in this last lecture, I'm going to simply talk about, so this goes back to biotech, okay? We talked about emerging separation processes, right, like single pass and disposable systems. What I'm going to talk here about is emerging therapeutics, all right? So there are a number of emerging therapeutics, and some of them require additional separation processes. For some of them, the current separation processes don't work that well. And so I think we can finish off by talking about them. I would say that in the development of these processes, you should think of it like this. So currently, if you're going to make a monoclonal antibody or some sort of protein, it's, I need to become a host again. It's relatively easy because there is all this knowledge, and all you can do is you can look at existing platform technologies, to polishing steps, so forth. You've got an idea of what you can add. Thank you. In the case of some of these emerging technologies, you have no idea, okay? So you're starting from scratch. You're basically the way people were maybe 30 years ago, 20 years ago, when they were developing technologies for protein-based therapeutics. Okay, let me share. And application. Emerging therapeutics. I think this is the one. Yes, okay. Yeah, very good. So that's where we are with this. So let's go back. So this is actually the very last lecture then. And let's just recap what we've done. So we talked about bioreactor harvesting, then introduction to membranes, sterile filtration, ultrafiltration, membrane adsorbers. Professor Titham gave this one on electroactive membranes, virus filtration, membrane biofouling you had a lecture on. I have talked about I changed this order a bit. So I talked about blood oxygenators, and I also talked about cell washing. And then I talked about leukodepletion, plasmapheresis, and hemofiltration. And then this one was given to you earlier, okay? And so then we ended up with these two lectures, and I changed the order here. Instead of talking about vaccines and so forth, I decided to change them and first talk about developing processes. So new forms of ultrafiltration, microfiltration, so forth. And I pointed out that in the biotech industry, it's actually same for biomedical industry, it's very conservative. So if you come up with this great idea that's a completely new membrane with new materials, everything, it's going to be very tough to get it accepted, okay? You will have, the way it will be accepted is if you can show like an order of magnitude or more benefit in terms of either product recovery, ultimately saving money, okay? So product recovery or cheaper cost or something like this. But you know, the reality is it's very unlikely you're going to find a process that's an order of magnitude better. You might find one that's 10% better, okay? 10% people are not going to change a manufacturing process, okay? They're going to say it's not worth it. I've got to spend all this money first to change the whole process and get it validated. And so what is my timeline for recovering this expense with a 10% cheaper? Secondly, I don't even know if your process works, okay? You say it works, you're giving me this data, but will it really work for my condition? I don't know, for my particular operation. And if it doesn't work, I'm in a lot of trouble because I had a process that works, it approves, and now I've done something else, you see? So it'll never happen. So that's why in all of these, in the last lecture, I talked about modifications of existing technologies, combining membranes with precipitation, right? Combining membranes with chromatographic resins. These are things that we know have already been approved. So the approval process will be easier. It is more comfortable for people in industry to say, well, both of these work, why shouldn't the combined one work, right? I have a greater feeling that this could work. The other thing that you need to do, though, before you can introduce a new process, is make sure that you have scale-up data, okay? So companies need to have modules that go up in scale. They need to have shown data that this works. But then the tricky part is any customer will come and say, well, have you got any data for commercial operation? And that is where there's a problem, because you don't. And so at some point, you have to work with somebody to commercialize it, and they have to be willing to co-invest. And that's where really the catch comes in, okay? So again, it's a tricky process, okay? That's why everything I talked about was modifications on existing processes. I didn't talk about trying to develop new membranes. Of course, we're trying to do that, but the timeline for that is 20 years or more, okay? So now let me talk about developing new therapeutics, because new therapeutics are going to require development of new unit operations. But initially, the way this is handled is by using existing operations and then trying to look at modifications of existing operations, okay? And so this last lecture then focuses on these new therapeutics or emerging therapeutics. And just one second. Okay, so the past 2 decades have been focused on monoclonal antibodies and monoclonal antibody-related products like fusion proteins, these various fragments, and antibody drug conjugates, and so forth. So, if you're a biochemist or molecular biologist, all this means something. If you're not, these are all protein-based therapeutics. Okay, they are using cells to produce proteins, fractions of proteins, parts of proteins. And so they have different names, fusion proteins, depending on how they come, but that's what it is. There's growing interest in commercialization of more complex therapeutics, such as live attenuated virus, viral vectors for delivery of gene therapy agents, plasmid DNA, glycoconjugate vaccines, cell-based therapies, etc. We have mRNA vaccines and so forth. So what they're saying is that, we might be able to develop therapeutics that make use of things other than proteins. Now, there are two strategies here. So one is, if the current way that we take these proteins is you can inject them, right? You can take them as tablets. You can drink them if it's an oral supplement. Sometimes you have inhalers if you want to get them into the lungs. There are various ways like this to go in, but they're not very selective particularly, right? If you inhale it in the lungs, true, it's going to the lungs, but it's going everywhere in the lungs. And so that's where this idea of gene therapy and viral vaccines comes in. You may want to just target specific cells in the body. And if you want to do that, it becomes a little bit complicated. So think of the classic example of chemotherapy for treating cancer. If you have a cancer, say, somewhere that you do chemotherapy, you try to localize it as much as possible, if that is possible. But nevertheless, you will kill off lots of good cells as well as cancer cells. And so that's why chemotherapy firstly has one reason why it has side effects. There's side effects for other reasons as well, but also why you have to be very careful with it, because if you give too much, you will end up doing more damage than the treatment's worth, okay? So the idea came up that gene therapy has been discussed for years and years and years, since almost the late 80s. For a long, long time, and it's a very nice idea. You take some sort of vector that will take your drug directly to the location you want to inject it, or where the cells are, and it'll attack just those cells. And there are various ways you could try and do it. In this work, we are focusing on virus particles. Now, there are synthetic polymers people have tried to use. There are many other methods that you could look at. Virus particles are very good for one thing. They are very, very selective. So they are very efficient at getting your therapeutic to the site you want to get it to in a very specific way. So that is the big benefit, okay? The downside is all sorts of other issues in terms of toxicity of the virus and various things, and purifying the virus. So I just want to point out there are people working with polymeric delivery systems as well. The problem is the efficiency is low, okay? All right, so then we have things like gene therapy. I talked about that. Cell-based therapies, and then also this idea of just using plasmid DNA, mRNA, things like this. So these are all much larger than recombinant proteins, okay? So we have already talked about virus filtration, where separation is difficult because the contaminating virus particles are similar in size to our product. If we're going to make any of these drugs by biotech means, we have that same problem. We have to remove contaminating virus particles. We might be trying to purify a certain virus, but there could be other contaminating virus particles. Those regulations remain. So we have to find a way to remove them. And now if our product is actually larger than some of these contaminating virus particles, our idea of size-based separation may not work, or it may have to be reversed, okay? So what we want is retained, and what we don't want goes through. But you can see that there's going to be problems here. Another major issue is significant diffusional mass transfer limitations in the development of chromatographic separations using traditional porous resins. We've already talked about this, right? We said that for large proteins, there is often a significant poor diffusional resistance. That's why membrane adsorbers are nice, especially in flow-through operations where you can adsorb the larger species. This is only going to get worse if you're going to use some of these species, okay? The other point is very simple, I think, also to understand. It's difficult to develop sterilizing grade filtration processes in which the product recovered in the permeate through a 0.2 micron size filter may be limited because it's getting to be rather big, okay? So if that's the case, or you might have imperfect passage, so you have significant rejection or retention. So again, there's a problem here when you think about sterilizing grade filters. So all of this means that there are a number of challenges in this area, and I'm telling you, these challenges are actually just emerging. This is where a lot of research for engineers anyway, it will exist. Okay, so let's talk about some of these then. So glycoconjugate vaccines, oh, I forgot one more thing, sorry. I mentioned here that there are live attenuated virus, viral vectors, there are virus-like particles. There are many, many types of virus vectors, okay? So you can use live virus, you could use killed, in other words, virus that cannot replicate. You can use virus-like particles. These are basically virus capsid proteins that have been formed in the shape of the virus, but everything inside is gone. There's no RNA or DNA, it's just what you put in there, okay? So there are many sorts of these things. So glycoconjugate vaccines formed by covalent coupling, i.e. conjugation, of the outer capsular polysaccharide from infectious microorganisms to highly immunogenic protein. And examples of glycoconjugate vaccines are tetanus toxoid or non-toxic mutant of this particular diphtheria toxin. So that's one type of vaccine that one can make. But again, you can see these are rather large species, so separations becomes complicated. Glycoconjugates provide much stronger immune response than polysaccharide alone, particularly infants and the elderly. Glycoconjugate vaccines developed for immunization against these various diseases that you can see here. So these are actually available commercially today. The critical step is separation of the glycoconjugate from any residual protein and polysaccharide that remains after the conjugation reaction, okay? So you're covalently bonding these two together, but you must remove all of the, if you like, in chemistry, if in simple engineering terms, any of the reactants. You've got to get rid of them, okay? And to very high degrees. You can't just say you removed, 90% or 95%. It really has to be very low, okay? Chromatographic processes, hydrophobic interaction, reverse phase, and size exclusion have low throughput due to mass transfer resistances, okay? So these are very big, and therefore they can't go into the pores, so you're only binding on the surface of the pores, so capacity is low, and there's significant mass transfer resistances. Also, poor selectivity, similar chemical properties of the free polysaccharide and conjugate product. So this is a very difficult separation, okay? There's interest in development of selective ultrafiltration processes that can exploit differences in size between large glycoconjugate and small protein polysaccharide components, okay? So this again gets back to something we were discussing earlier. Ultrafiltration is nice. It's an existing technology, right? We've got plenty of data on this. It's used, it's approved, everything is done. And as somebody pointed out earlier, yesterday or the day before, we use ultrafiltration for concentration. We use it for buffer exchange. We don't use it for fractionation of anything at the moment. But it could be used, right? And then I discussed this idea of high-performance tangential flow filtration, and we talked about trying to separate things that are similar in size, oligomers and monomers, right? Monomers and dimers, and so forth. So maybe this is a very good application for high-performance tangential flow filtration, right? Potentially, it could be very good, maybe much better than chromatography. But here's a caveat. We talked about HPTFF, and actually the first papers on HPTFF came out now, must be well over 10 years ago, maybe 15 years ago. But if you go around and look for any industry that's using high-performance tangential flow filtration, I can't say there aren't any, but I'd say you'd have a tough time finding any, okay? So very, very slow commercialization. The question is why? I mean, this would be an obvious thing. It seems very attractive, right? So here's the point. It had not had a significant impact on commercial processing of MABs, monoclonal antibodies, or antibody-related products due to challenges in displacing well-established chromatographic processes for antibody production. In other words, you're never going to put it into an existing manufacturing facility where things are working okay. I mean, you may go and make an argument and say, if you put this, you'll get 10% more profit or something like this, okay? But that's not going to buy anything, because what people will say is, well, yeah, that's very good, but we also have to shut down for a while. We have to get the whole thing revalidated. That'll cost money. So where is our return on investment? I'm not going to take the risk, okay? Then the idea is, what about a new manufacturing facility? Yes, now you have a chance of bringing it in. But again, for monoclonal antibody production, we have methods we know that work well. We know they're like platform technology. So why do I want to try something else when there is the promise of only 10% improvement? Okay? So that's the reason it hasn't. But there are certainly possibilities for some of these developing areas where we can't. There's a niche application. Current technology won't do it. Okay? So that's where these sort of things come in. Another challenge in downstream processing of glycoconjugate vaccines is the final sterilizing step. And that's obvious, right? At the very end, you have a sterilizing step. And what happens if your product is too big? Now, it's not larger than the membrane pore size, but it's getting pretty big. So you might have significant rejection. What are you going to do, right? You're losing your product. So again, are there alternative ways that we could run this final sterilizing step? Are there ways that we can get a better differentiation or separation between things that are very close in size and very close to the pore size? Okay, that's another big challenge here. Glycoconjugates are 100 to 300 nanometers in size by dynamic light scattering, comparable to the 200 nanometer pore size in a sterilizing grade filter, right? So we have a major issue here. So sterilization is a problem. It leads to significant fouling. So these are some of the challenges if you end up working in this area that I think will be in there in the future. So use of DNA or RNA for the replacement, inactivation, or modification of one or more genes is another very interesting area of development, okay? So the early focus of gene therapy, so we talked about gene therapy a little bit. The early focus of gene therapy was treatment of hereditary diseases characterized by a single missing or malfunctioning gene. The introduction of gene that codes for the missing gene, say a missing clotting factor, for example, factor 8, is a treatment for hemophilia. So you're genetically missing this gene. If I can somehow find a way to insert it, right, and that's great. Then you overcome the problem. However, there are other ways where you can look at replacement of or inactivation of just DNA or RNA units. Okay, that's one area of development. The other is it possible to use gene therapy for treatment of cancer using genetically engineered T cells. So basically, you're trying to kill off these T cells, right, or these cancer cells by doing a mutation that is genetic in nature. And then because other ways, the thing is cancers tend to recur. And that's because you can never totally kill off all the cells. So DNA and RNA can be used as vaccines. We've heard about that recently. The nucleic acids provided for the in situ production of target antigen, which then stimulates the patient's immune system. So you don't have to give the whole virus, okay? You just give a DNA or RNA that stimulates your immune system to start producing antigens. So all of these are interesting and developing therapeutics, okay? They've all shown to have therapeutic effect. In the upscale area, people, microbiologists have done tests. They show that this is all of great potential and possibility. The question is, can you do it commercially, right? Can you grow them? Usually the upstream problems are not that severe, but then can you purify them so that when you end up with a drug, it actually has more therapeutic effect than side effects, right? You don't want it to have side effects, then it's not worth it. Okay, so here are some of the things that can be done. DNA delivered to cells of interest using viral or non-viral vectors. I told you virus vectors we are talking about here, but there are non-viral vectors as well, okay? Non-viral vectors include a range of polymers, peptides, nanoparticles, and lipids. There are advantages because you don't have all of the possibilities of say, having to validate clearance of host cell protein, DNA, these sort of things, but they're often not as efficient, okay? So you're not actually getting the genetic material into the cell that you want to efficiently. DNA in the form of a plasmid, a small extrachromosomal DNA molecule. Plasmids exist in one of three topological isoforms: supercoiled, linear, or open circular. Okay, this is quite interesting. So a supercoiled isoform generated when the plasmid is made through enzymatic winding of DNA. The form has the greatest therapeutic activity. So actually, supercoiled plasmid DNAs is what you want. okay, typically. Open circular isoform generated by breakage in one of the DNA strands referred to as nicked, and the linear isoform is produced when both DNA strands are cut, okay? So you have these three forms. Linear and open circular isoforms are processed impurities. They are considered impurities and must be reduced to less than 20% of the total DNA according to FDA guidelines in the US, and it's going to be the same everywhere else, okay? So when you produce these, you will have a range. You need to get rid of these other two. So how do you do it? It's a challenging separation given the different DNA isoforms have identical number of base pairs and base sequence. What is different is their shape, right? Supercoiled, linear, and open circular. So how would you separate them, right? So a chromatographic separation is going to be very difficult because if they all have the same sequence and base pairs, what are you going to use to differentiate them, right? There's nothing. Size based on the surface of it would seem difficult because they're all about the same size, right? So what can you do? So here's an interesting idea. Look at this. So a membrane system is attractive for purification of these plasmid DNAs, and the reason is that tangential flow filtration could be used for initial purification of plasmid DNA. Host cell proteins and other small impurities are removed in the permeate. So that's obvious, right? So you're going to get the plasmid DNA, you're going to reject it and remove host cell proteins, host cell DNA. You have to do that anyway. So now you have this plasmid DNA, but you have all three isoforms. And the question is, how are you going? This is a new separation challenge. You don't have this with protein separation, right? This is a new separation. How do you separate them? And so one interesting idea is this one. So transmission of large plasmids radius of gyration between 50 and 200 nanometers through relatively small pore ultrafiltration membranes, pore size 5 to 15, due to elongation of the plasmid DNA in a converging flow field approaching the membrane surface. So what this is trying to show you is that you can make use of the flexibility of these different species, right? So that in this, but you have to have this converging flow field where you can see this starts to elongate. So it can actually pass through. So DNA elongation, a converging flow field at the intramembrane pore, left panel, okay? On the left panel, you can see. I'm sorry, this has got cut off. So the low flow rate, low filtrate flux, right panel is high filtrate flux. So at low flux, it's not going to go through. At high flux, it could go through, okay? So what this suggests is, but this is for this particular isoform of the DNA. So what this suggests is that by again carefully designing our ultrafiltration system, but more than that, the flow field, we may be able to separate this. Okay. So these are new separation challenges, not relevant for proteins, but become quite important in this area. So what can we say then? Sorry, this has to move on. Okay, so basically what we can say is this. All of these ideas are stuff we have talked about already. The critical flux for transmission is different for supercoiled, open circular, and linear isoforms due to differences in their elongational flexibility, right? So you have this converging flow field, but one of them may go through first. In fact, there's a linear form which may fit through this much more easily. You have to have higher flow rates to get the supercoiled form to squash into this and elongate, right? So linear isoform is the lowest critical flux and most easily stretched and aligned with the flow streamlines entering the membrane pore. The open circular topology has the largest critical flux due to bending stiffness of the expanded ring structure. So one could hypothesize then a separation where you have these three isoforms. You first run it at low flux and you remove as much as you can of the linear isoform, right? And then you run at a higher flux, maybe, and try to recover as much as you can of the isoform you want while rejecting the isoform you don't want. Okay, that's one way to do this. So you can see that, but all of this is going to require very, very careful design of the membrane pores, operating conditions, and buffer. And so these are ways people are looking at to try and do this, because it's a completely new separation problem. Okay, let me move on then. There are a couple more things to talk about. So if ever any of you decide to work in this area, I suspect these are the sort of areas that would be of greatest interest in the future. So another one is viruses, virus vectors, and vaccines. So early application of live virus as a therapeutic was actually the development of smallpox vaccine, and I just learned this recently. It was a long time ago, 1796. It was actually even before the actual virus had been isolated, so that was quite amazing. But a cure had been found, or a treatment rather, for smallpox had been found that depended actually on a vaccine. So vaccines are based on live attenuated virus. So we have vaccines for measles, mumps, and chickenpox. That depends on a live virus that's modified a bit, so it infects you, but you don't get the full infection. You get a partial infection that then change slightly because the influenza virus is changing. And it's actually a concoction of about 6 different vaccines all put together. And then you take that. And so this is actually an inactivated virus. Okay. There are virus-like particles, and these are used for hepatitis B and these other diseases. So as I said, virus-like particles are clever. What you do is you set up your whole bioreactor where you have the capsid proteins and you get them to fold into the virus capsid. So the cells are producing the capsid proteins, but there is nothing inside them. They're empty capsids. So there are no viral DNA or RNA. So they're actually totally non-pathogenic. That's the good thing about them. So you could use them in a low biological safety pattern because they're totally not going to affect anything or do anybody, any damage to anybody. The point is that I mentioned earlier, when a microorganism produces virus particles, it doesn't produce all active particles. It produces active ones, partially active, defective particles. So this is just taking a time to just get to produce only the defective ones. Okay? And that can be done. First, oncolytic virus, genetically engineered form of herpes simplex virus, okay, that can kill a number of tumor cells, but food for treating cancer and viruses. So this is a virus that was designed to go and attack a cell to stop tumor. Okay? And finally, virus particles based on adenovirus, lentivirus, and associated virus, AAV, are studied as virus vectors for gene therapy agents. These are probably the most advanced, adenovirus, lentivirus, and adeno-associated virus. Okay? So you can see that today there are a great deal of virus particles that have actually already found applications in industry and are already representing commercial products. But all of these, many of them, especially the gene therapy ones at the bottom, are rather expensive. And that's because the efficiency, one of the reasons of purification is very, very low. Okay? So let's look at what we're trying to do here. So AAV, adeno-associated virus, are attractive for targeting neurological disorders like Parkinson's disease. And the reason is, and Huntington's disease, due to their small size. These are very small virus particles. They're actually similar to parvovirus multivirus of mice, about 18 nanometers in size. And enhanced safety arising from the need for co-infection with helper virus to achieve effective replication. So the problem with this is that we don't want to go and infect a patient and the patient starts producing more virus. And associated virus is an interesting virus. It cannot reproduce by itself. It needs adenovirus to reproduce. Okay? So there's that symbiotic relationship. So that's why they're relatively safe if they're by themselves. Okay? So this thing called Luxturna, the first FDA-approved gene therapy treatment for inherited retinal disease, is delivered by the patient. So what's interesting here, AAV is actually a very nice vector. It's relatively safe compared to other viruses. The problem is, it has taken years. This first one was approved at very recent, 2020. And the reason was people couldn't purify this virus, and so the side effects were very serious because we had a whole circle, this is a big issue. Today there's a second one also. I think there are two AAV therapies. Oh, by the way, we wrote AAV now as part of our work on virus purification. And it gets more complicated because AAV, there are many forms of AAV. There's AAV 1, 2, 3, 4, all the way up to 7. Just slight changes on the AAV. AAV 7 and 9 are the ones that are used mainly for therapeutic applications at the moment. Certainly, viruses produce either an appropriate host organism. So, human influenza virus used to be produced in chicken eggs. Today, there are cell culture. Before, the best way was chicken eggs, which is really not a very scalable process and rather complicated, right? But anyway, chicken eggs for measles or human influenza virus, or by cell culture, at least for hepatitis B vaccine, to embryonic kidney cells, cell lines like this. So production of the virus is not something we're talking about in this course that much because we're doing downstream purification, but that is also complicated. So, okay, so I think until about 2010, most human influenza viruses produced in chicken eggs. So there was a big problem because this took forever to do, it was complicated, you couldn't suddenly reach peak loads because the growth of the virus was complicated. There are cell culture ways to do it. Viruses range in size. This is what is another challenge, from 20 nanometers to 1,000 nanometers. Okay? So 1,000, the measles virus is about 1,000 nanometers. Challenging throughput downstream purification processes compared to proteins, right? Much more challenging. Additional steps required to release virus from the host cell, chemical lysis or modulation or sonication, often necessary to remove incomplete unfilled virus capsids. So there are two steps here that are very important. So if you remember, we talked about bioreactor harvesting, and we said that actually here, in most cases, for therapeutic proteins, except perhaps in bacterial cells, they are excreted in the growth medium. That helps a lot, right? Then have the protein of interest in the growth medium, the cells excrete the protein, and then your first purification step, the harvesting or purification step, is removing particulate matter. Most viruses, in fact, I don't know an exception to this, but there probably is, so I won't say that more. But most viruses, the virus particles, the majority of them are inside the cells, okay? A few are excreted through, but the majority are inside the cells. So for this to be an efficient process, you're going to have to lyse these cells. And lysing, whether you do by chemical lysis, you can use a chemical way to rupture the cell membrane or make it more porous. If you use homogenization and phosphonication, you smash up the cells. Whatever you do, it's going to be complicated because now you have all this cell debris floating around. If your virus is fairly robust, like AAV, you can still separate it because even though you've released all the contents of the cell, the enzymes, and all sorts of other things, they are still fairly stable. If you have a very fragile virus like measles virus, which is rather large also, it's 1,000 nanometers, the fact that you have all these enzymes and things floating around actually often affects the virus. And if you lose the surface markers on the virus, then it's useless for any therapeutic because it's gone. Okay, your body doesn't recognize it. So that's a major additional challenge, which is additional separation step. And the other one is to remove unfilled and filled capsids, which is something we are working on a lot these days. I told you that when viruses are produced by host organism, they produce the actual virus you want, filled capsid. They produce partially filled capsids, defective capsids, non-infective, that's all fine. They produce all this. And in nature, that's fine. For some of these viruses, the ratio of infective to defective is like 1 infective for 10 defective, so it's very hard. Okay? This also explains why some viruses are more effective than others. Okay? And because you maybe taking a lot of defective capsids won't do anything. Okay, so some viruses are more effective than others. So, from a therapeutic effect point of view, you want to get rid of all these defective capsids because they are not going to have any therapeutic effect, but they have the same surface binding markers, so they'll go and attack the tumor cell, whatever it is, but they won't do anything. Okay, so you've got to remove them. So how do you remove them? So think of it. Here's the separation. You have the viruses that are defective and filled, but what's the difference? Well, there's a very slight density difference, but not very much. Okay? It's very slight because this one doesn't have much DNA or RNA, and this is a full copy. The surface is exactly the same. This one, the defective one, might have a slightly different isoelectric point because DNA or RNA is charged. And so the defective one doesn't have that inside the capsid. So there might be a slight difference. Similarly, there might be a slight difference in hydrophobic. But these are slight differences. But the other problem is you don't have just filled and defective. There are various forms of defective, right? You might have 20% of your DNA, 60%, 50%. So there's a range of isoelectric. And so this turns out to be a very, very complicated separation to separate the filled and defective capsids. That turns out to be very, very complicated. I can take one of the things that's done today. Today, people use resin-based chromatography. They typically use an exchange try to, based on differences in the aspect, try to separate them. But often the recovery of what you want is only about 50%, which is really unacceptable in the market. That's why they're so expensive. Something that we are working on, which is of course highly researched at the moment, is to design multimodal ligands on membrane absorbers that could basically help with the purification, say by binding based on ion strength and hydrophobicity. And by trying to combine both of these, we hope that we can get a better purification. But anyway, that's some of the those are two major additional steps that are complicated in this. Okay. Next slide. So this is some examples of viruses. So another problem is sterile filtration. So look at these. These are some of the viruses that are of interest. So there is okay, so let's look at this. So we have here adeno-associated virus, the gene therapy application, about 20 nanometers in size, shouldn't be too difficult in terms of sterile filtration, right? They're pretty small, and they're grown using insect cells and these human embryonic kidney cells, and they also need adenovirus development, otherwise it can't be it's fine. Adenovirus, you can see here, they're about 90 nanometers. They're used in cancer gene therapy, human embryonic kidney cells. 293 is the cell line, the same cell line, which makes sense, okay? 90 nanometers. So if you think of 90 nanometers, 0.2 micron core size, so that's 0.49 micron. It's getting a little tricky, right? It's getting closer. But let's look at some of these. This one, which is the herpes simplex virus that's used for cancer treatment, it's an oncolytic virus. Or you look at, say, measles virus, 1,000 nanometers growing chicken embryos, and for that matter, even influenza virus, 120. They're getting rather large, okay? And this one, measles for sure, sterile filtration won't work. And then lentivirus is close. You should be able to get lentivirus. So I told you the most promising ones today are lentivirus and adenovirus, those three. And for those, we could probably get away with current sterile filtration approaches. The others, it's going to be very, very difficult. So you can see that sterile filtration is critical throughout the downstream processing stream. It occurs everywhere. And so here's another challenge, okay? How would we do this? You can't gamma irradiate or do it too bad, right? We've got to come up with better memory processes. Okay, a few more things. Sorry. Okay. So the first of these antigen receptor-based T cell therapies was approved by the FDA in 2017. Therapeutics use patients' own autologous cytotoxic T lymphocytes collected and genetically engineered to express an antigen-specific receptor that enables the cells to recognize and kill target tumor cells. So what's interesting here is that this is basically a cell therapy, right? What we're doing is the cell-based therapy. We're actually taking cells, your own cells. It's tricky to take someone else's cells, especially when they're white blood cells. But take your own cells, modify them, right? And then reinfuse them to particularly go and target tumor cells. That's very nice. I mean, conceptually, that's very nice, right? You take your own white cells, so you're using your own immune system, you're helping it to go and kill tumor cells, right? So from a conceptual idea, that's a very nice way to do it. That's what's happening, okay? So the RNA sequence for these chimeric antigen receptors, which is a fusion protein containing the antigen binding domain for monoclonal antibody and specific domains for T cells signaling, is transferred to the T cell using a lentivirus technique. So here is where virus purification comes in again. Okay. Now, production of this therapeutic begins with collection of the T lymphocytes. Now, we've talked about blood processing, right? Blood fractionation. So here's an example where we actually want to collect the white blood cells from a patient, but for your own use, not for somebody else, okay? From a patient's body by selective centrifugation, something called immunophoresis, okay? I didn't talk about it, but we talked about blood processing, because today this is really a centrifugation process, not a membrane process, okay? T cells are collected in a fraction between the more potential red blood cells and but actually, if you take a blood bag like this and you centrifuge it, you will end up here with a very, very red region. So these are red blood cells. And this typically is about 35%, 30-35%. And then here, you end up with what should be yellow. There may be some parts, basically yellow. That's all your plasma proteins. And in between the two is something called a buffy group. Okay, it looks a more buff color. brownie color, and that contains platelets and white blood cells. Okay. So that is how, and then leukapheresis basically you take this graphicode and then you do further centrifugation of that to try and recover your lymphocytes and ions. Okay? Okay. Let's move on. So, after you collected these T lymphocytes, next T lymphocyte activation occurs using antibody-coated magnetic particles, these dynamics or nanobeads, to facilitate genetic modification. So basically, what you do is you take the T lymphocytes. These beads are sold commercially. They have various, if you like, ligands on them that specifically go and bind to the T lymphocyte, and therefore you can add something to the surface and modify it. But we are using magnetic nanoparticles. So the way you separate is once they bind, you put a magnet and pull them on out. That's how that works. So T lymphocytes are then genetically modified, expanded in the appropriate bioreactor, they're modified with antivirus, they're expanded, they're grown some more, concentrated and formulated for cryopreservation. Indeed, we talked about cryopreservation and the lymphodepletion. So you can see that anyway, whatever you do, this is going to be a very expensive therapy, right? Because there are many complicated steps. But these are some things of interest. Now, interestingly, use of human mesenchymal stem cells for repair and regeneration of various tissues, cardiac tissue for people with heart disease, pancreatic tissue for people with diabetes, another area of interest. And a third one then is this one here, which I think I touched upon already. And the hematopoietic stem cells obtained from bone marrow have been explored for treatment of Crohn's disease and other things. Okay? So these are all taking human cells and trying to expand them. Okay? So it's cell-based therapies. So a number of possibilities exist. Number of possibilities for membrane separation through reduction of cell-based therapies. So one is this so-called LOBO, local cell processing system by Fresenius, designed for concentration and washing of harvested red blood cells. It's a membrane process. Novel pre-sterilized single-use microfiltration device with large pores. The pore size is about 4 microns. Okay. Using here the example we talked about yesterday, it's a crack-edge membrane. Remember I said crack-edge membrane usually not used, they don't want it. Here you want them, okay? And actually you're not collecting that many cells, so it works. Crack-edge membrane for separating different size cells. Membrane is wrapped around the inner cylinder of a spinning and membrane filtration unit. Centrifugation, referred to as Taylor quid, to Taylor vortices in narrow gap between the and outer cylinders. Taylor vortices increase mass transfer, reducing fouling, and you can remove, this is what's interesting, 98.8% of platelets, 2 to 3 micron in size of the permeate, more than 90% recovery of CD34 positive cells. Those are actually the genetic hematopoietic cells in a two-step washing process. So I'm not sure if I have a and so I say here, temperature flow filtration using 24 micron pore size followed by the polysulfate membranes used for concentration of mesenchymal stem cells. So here's what we do. I think the battery is low, right? Okay. Now it's on again. Okay. All right. Thank you. What was I going to say here? So here's the system. Okay. So you have basically a membrane here. You have this thing rotating around here, and then these Taylor quid vortices. Some of you may know about. It's a vortex that helps increase the rate of mass transfer by using this. Okay. But the aim is this: you want to have your feed come in here. Okay. The feed comes in here. Larger cells that you want to recover, say, will come through here. Smaller ones that you want to recover permeate will go this way. Okay. But it wouldn't happen if you just ran tangential flow microfiltration because the sizes are too close and you won't get efficient separation. But by having the spinning filter and designing carefully, you can actually get this. So this is the schematic diagram of this local spinning membrane cell processing system. The smaller yellow cells represent platelets, while the larger orange cells represent stem cells. That's one thing. I should point out that this is one application. There are other applications for these microfiltration membranes. So I think they're almost at the end. Let me finish one or two more slides. Okay, so cell-based therapy sample. Very large pore size, normal flow filters. This is normal flow, 30 to 100 micron in size. Used for recovery of human mesenchymal stem cells after detachment of the cell from large microcarrier. used for GIAN culture. Nylon filters used to recover human animal-derived stem cells harvested from fat tissue based on strong absorption of stem cells in nylon membrane. Cells recovered by reverse flushing with provide serum possible to select for cells with high ability to differentiate in bone marrow cells in the thin membranes in which nylon was coated with a layer of recombinant but collecting glycoprotein cells. Adhesion membranes system attack the point that can remove a product cases like this some outside. So this one we've talked about a lot. Okay? I think it's that we've got again. Should I wait? I can wait. We've almost got 2 slides left. So let me just give you some other background information in the meantime. So what you can see is that there is actually a huge area of development for cell-based therapy. This is quite different to proteins, viruses, vectors, or it's actually using cells, your own cells. Okay? But when you start to use your own cells, you need to be able to recover them. And that's one example. But there are many other examples. You know, another thing that people are working on a lot is various sorts of cancers. They like to go and try to recover the metabolic stem cells. So if you didn't have your cord blood and froze it, we still do have progenitor cells in our bodies, but as we get older, the number decreases. But we do have a few. So one could try to recover them. Okay? But in doing so, you often find there are circulating tumor cells as well. That's another separation like this that is quite interesting and complex. Okay, so basically, this last one we talked about already. I told you about cell washing. All of these are talking about basically ways you can use these nylon filters where you selectively try to capture the cells that are the ones you want, that are targeted for whatever you want to do, whether it's developing various more immune system or whatever. So these cell-based therapies are a great growing interest. Okay, next. Membrane device that combines tangential flow and normal flow filtration while exploiting cell settling to minimize membrane fouling. So this is an interesting device. 0.65 micron pore size PVDF membrane is used here. Okay, it kept free of cells which tend to settle at the bottom. So you have this thing here, and the cells being more dense tend to settle at the bottom. Okay, this is your 0.65 micron pore size PVDF membrane. Okay, cells are concentrated by pumping a small amount of air inlet to force the fluid to the membrane. So you can pump a small amount of air here, it forces the cells to the membrane. You can get more than 85% removal of DMSO with high recovery of bone marrow derived human mesenchymal cells. I thought this was interesting because it's an example. You've got cross flow, but then every so often you change it to dead end filtration or normal flow filtration. So that's an example of the type of things, again I was pointing out, in the future we're going to look at combining these and coming up with enhanced or unit operations where you combine more than one separation process. Okay, membranes for cell fractionation. Membranes for cell fractionation based on differences in size and deformability. Separate circulating tumor cells from blood cells. I referred to this a moment ago. Smaller red blood cells and lymphocytes wash through the membrane for permeate recovery. Dead end filtration through 3 micron pore size tracking. Manufactured red blood cells. So again, one can look at trying to fractionate cells, and membranes play an important role here. Okay, and we make use of cell flexibility as well as size and so forth, and deformability. Okay, future directions. This is the last slide. So basically, if we think of membrane separations, this emerging therapeutics that are non-protein based is going to be a huge area of development. Okay, particle-based delivery systems. I talked about virus vectors and others, and that's a large area as well. It's working. Anyway, never mind. For the last 2 minutes, okay, we'll put this on again. Particle-based delivery systems I talked about, but perhaps the most interesting is going to be these cell-based therapies, because actually that is really a future development. And ultimately, if you can come up with therapeutics where you use parts of your own body cells from various parts, that's going to be the safest. Okay. Additional purification steps often required. I talked about these in terms of trying to separate filled and empty virus capsids. I talked about them about trying to get virus particles from outside the cell to come out, and then obviously cell-based therapies. Existing processes may not be suitable for large size of particles compared to protein. So that's the major take-home message there. So I think that gives you a summary of, that's the end. I think that gives you a summary of not only some of the emerging technologies, but also some of the emerging therapeutics, because ultimately the technology to develop will have to be driven by what the therapeutics are, right, if they're to be feasible. So I think that's it for me. What happened is Dr. Jacob back, or I saw him a moment ago. Did he leave? Okay, I finished. So he was going to come now and give out your certificates at some point. You want to call him or see if he's there? Okay. Any questions on the whole thing? Any questions?
Sir, if we use cell-based therapies,
yeah.
So that would be much, if you use membrane process for separation, it would be like more pure. If you use that, we can have more purer form of cell therapeutics compared to a gene therapy or a virus.
Yeah, because you are using your own body's parts. And so you get rid of all of the possibility that host cell protein, host cell DNA, these sort of things tend to disappear because you are using your own cells. So yes, absolutely. But the problem is trying to recover them, capture them, you've got to expand them, and so forth. So there are a lot of processing steps. But yes. I mean, cell-based therapies have been discussed for quite a while. They're not new. I mean, people have been talking since the 90s. That's when people started freezing cord blood from the umbilical cord. But at that time, it was a matter of, well, maybe we just freeze this and keep it, because in 20 years' time, there may be a therapy, and by the time you're 50, 60, you may need it. So that was the reason. But now some of these therapies are appearing. So, but they're very expensive. Okay. So there's, I mean, I just showed you the number of processing steps. Right? So, okay. Very good. Any other questions? Yes.
So how the membranes can specifically selectively separate the effective and defective viruses? Because there is a very
yes. So the hypothesis is this: if you use a single mechanism of action, just say charge, it's very difficult, because what I wanted to show you was if you just use charge, where did I put the pen? Here. You have the following problem. So if you do a chromatographic separation, okay, what you'd like to do is have an elution peak like this, okay, and you elute this by changing the pH of your buffer, and at some point they'd come out, okay? But now you have things with slightly different isoelectric points. What you'd like to get is two peaks like this. So this could be, say, a defective, and this could be the filled capsids, the ones you want, right? But in reality, you get a peak that looks something like this, and then like this, and like this, and like this, because you have all sorts of partially filled capsids. So this separation, and you can then take these and do an infectivity test on this fraction and on this fraction, and you can show that you have more infective particles here than here, and therefore show where the the filled capsids are coming from. However, the separation is not practical, okay? So the idea is, can you also then use hydrophobic interaction as well, so you try and get the spacing between these two peaks increased, right, and try and recover. Now, the reason for using a membrane is because for a chromatographic resin particle, due to resistances, pore diffusion resistances, these particles will not bind on the inside surface at all. So your capacity is very low, and you have all the problems with binding large particles. But that was the idea, to use multimodal ligands, to use multiple mechanisms to try and target the ones you want. Did I answer the question?
The multimodal ligands that we incorporate the space, how can they exactly, what's the way?
What do I mean by multimodal? Or
The surface modification that we have done.
Yeah.
How can they exactly differentiate between those two effective and defective?
Oh, because it's like this. So if you have a certain, if you have the isoelectric point, the net charge on the virus will be 0. So you won't have a charge-based interaction, it will elute. If the isoelectric points are different, you do a pH gradient in your elution step, and as you pass through each isoelectric point, you start to elute. But the problem is these peaks are all overlapping, so they're trying to separate the peaks. But yes, that's how it works. Same with hydrophobic interaction. So you can have phenyl groups and other things, but as you change the salt concentration or the ion strength at different points, their binding interaction will be less, so they will be released. So that's it. Other questions? Anybody? No? Okay, so I think I am done. No more questions. We're finished.